Okay, so in this lecture, we'll talk on the multiplex PCR. The multiplex PCR is the PCR where we use the more number of primers to detect the large number of genes in the genome of a cell or of a microorganisms or pathogens. So the advantage is that when we have a large number of primers, maybe more than 2, 3, 4, 5 primers, then at a time you can have the primers. Now, one of the challenging tasks with the multiplex PCR is that the optimization of the reaction mixture, because if you have 5 primers, if you have 5 set of primers, then you may have like chances to different conditions of these primers in different time period. For example, one of the primer set may have different annealing temperature to the other, and the other one may have varying temperature to the other one. So having a difference in variation in the annealing temperature could have one of the challenging tasks for the multiplex PCR. Now let's see, use multiple primers pairs in one reaction. Say for example 2, 3, 4, 5 primers. It requires the optimization of the annealing condition, as I have just told you, because if the annealing conditions are not optimized, so for that you need to have the gradient PCR, right? We call the gradient PCR. Now this gradient PCR will help you to give you the idea about the variation in the annealing temperature. So for example, in one of the set of primers, they have an annealing temperature of 45 degrees centigrade. The other one may have 50, the third one may have a 55, and so on. If the variation is increased, then you have to optimize between 40 to 50 degrees centigrade temperature and gradient, keeping all the primers together, right? So this is how we call the optimization of the temperature. Now what is risk? The non-specific product and uneven amplification might take place, because since you have a number of primers, and sometimes these primers might bind to a non-specific sites and give you the different products. So in that case, if you have a different, because you are expecting the large number of the product, right? More than one product, which is unlike to the specific single set of primers, you are getting single product. So in case if you are getting the single product in a single simplex primer, then your reaction is perfect. But if you are getting more than one product or two product, then you have it, you already know that this is not correct reaction. You have to change the reaction again, optimize the conditions, and you try to get the best possible product. But in this case, what is the risk? That when you get the multiple band, you might confuse whether it is because of the primer binding or because of the non-specific binding. So this is one of the disadvantage or one of the challenging tasks for this. Now, the risk factor is non-specific product production. Hot start and optimized buffer crucial. So hot start means like, say for example, you are not supposed to add the enzyme. In hot start reaction, the enzymes generally is not added, and enzymes is added later on, right? Enzyme at the later time means like when you add the enzyme when the reaction, first reaction, before reaction, the denaturation takes place. So once the denaturation is taking place, then you add the enzyme, right? So then we call hot start optimized buffer crucial, which is very important. The commercial kits recommended. So generally commercial kits are present. You can have DNTPs, you can have enzyme or reactions are there in a kit. What is important that you have to just optimize the concentration of primers in each set of reactions. So you have a multiplex PCR kits are also available. Now what is nested PCR? Nested PCR is a very important PCR where you need to have kind of maintain the specificity of PCR. Now generally in PCR, what happened when you run the PCR, see when you run the PCR, say for example this, you bind the primer here. So this is a 5' end, this is 3' end, this is 3' end, this is 5' end. 5', right? So primer bind here and primer bind here. Now when you amplify it, you will get this, and then you will have like this. Now these two products are formed from this. Then again you further amplify this one, and you can have again 4 products are formed. Correct. So this is how the amplification starts. Now, what happened with this normal PCR, non-nested PCR, where the nested PCR is not used? With this primer, which is giving you the product, this could have a non-specific product, but you are not known because you are getting single band, right? So in this case, you're not sure. So what the nested PCR will do, are even if you are getting more than 2 bands or more than 1 band, which is supposed to get a single band, but rather than to get a single band, you are getting more than 2 bands or more than 1 band. So again, you got a non-specific binding. So in order to get the specificity, what is being done, rather than single set of primers, 2 set of primers are used, right? So in case of a 2 set of primers, for example, first PCR outer primers, then second PCR with the inner primers. So suppose this is a product, right? You are getting one primer we call outer primers. Then you may have some inner primers. So this band to be of, say, for example, 1 kb, right? This band could be of, say, for example, 500 base pair or 0.5 kb. Correct. So what is happening in the nested PCR? First, you run the reaction with this, right? And whatever product is being opted, because this is the whole genome reaction you are taking place. For suppose you are getting this through genome. So you can have like a whole genome. When you use this genome, somewhere it bind here. Suppose this is the site of the gene. It bind here. It will give you this product, right? Or it will give you finally this product. This product, if it is correct product, right? If it is a specific product, then the inner primer that you have designed within the gene right here and here, this one, would definitely bind here, right? And if it is not a correct product, say suppose if it non-specifically binds somewhere here at this site and it give you some different product, then this inner primer which you have made on this amplicon to amplify to get the product will not bind here, right? Because they are made for this gene and you amplified this gene. All right. So what happened? You have to have inner primer, and if you use this product using this primer, then you will get the correct product. That small size product out of the amplicon you use. So you have the large product, large genome DNA, get the sequences and get them product, and after that you get whatever product you are getting, use the same amplicon product, use the inner primers, and then you try to amplify to cross check if these inner primers are belonging to the same product or not. And that is what the nested PCR works. So this is how the whole figure, you can go into detail and see the figure how it works, as I have explained in detail. So in nested PCR, the specificity has increased. The specificity of the primers are increased, and the specificity. So this is called traditional PCR, nested PCR, and this is the semi-nested PCR. So again, in the semi-nested PCR, you can have the cross inner primers, and you can have a target. You can amplify the target. You can have one primer inner primer, one primer outer primer. So suppose here you can have only 2 set of. Suppose if you cannot make a 2 set of primers, so you can take only one set of primers. In this case, you have a 2 set of primers, but here you can make 2 set of primers in the way that you can have 1 set of primers remain same. For example, outer primers and 1 inner primers, right? So you just make a 1 inner primer. So you do not to have 2. So you have, instead of 4, you can have a 3 primers, and here you have a 4 primers, and but you can have a little large size product compared to this, because here you can have small size product because you have make quite inner. So this is called traditional nested PCR. This is called semi-nested PCR. Now, I think the take-home message here is that the semi-nested PCR is basically, and nested PCR is basically important for identifying the specific gene product in the genome or the plasmid. Now, what is touchdown PCR? Touchdown PCR, uh, touchdown PCR is initial PCR where you can have the initial annealing temperature higher than Tm, 72 degrees centigrade. Uh, then you can have 1 to 2 degrees centigrade decrease per cycle until target temperature is to 50 to 60 degrees centigrade, which enhance the specificity and reduce the false priming. Now, this is the idea to again increase the efficacy or specificity rather, to increase the specificity, because if you're using the low temperature, like for example, if you're using the annealing temperature of like a 50 degrees centigrade, the primer may bind to different sites. But if you are initially the annealing temperature go to 70 degrees centigrade, the primer will bind like here. So this is the initial temperature like Tm plus 10 degrees centigrade. You got a denatures and extensions, and then cycle 15 up to 15 cycles reaction. Then next you can have uh, reduced temperatures, and then you can have next 25 cycles, right? And the extension temperature is 72 degrees centigrade, and this is the Tm. So the idea is that how you can, over a number of cycle move, you can reduce the temperature. We call it touchdown, as the name itself indicating, touchdown PCR. So touchdown PCR basically help us to increase the efficiency by reducing the temperature. And this has been done when there's a no kind of a so product is not formed and annealing is not taking place. So in order to get the annealing, you keep lowering the temperature, and starting from increased temperature, the idea is to increase the specificity by not binding to non-specific site at high temperature, because the probability get reduced. Now, the digital PCR is okay. The digital PCR is same thermal profile at PCR 95 degrees centigrade to 50 to 65 degrees centigrade, up to then 72 degrees centigrade is again extension. The sample partition into thousands of micro reactions. So this is the special digital PCR is a special machine used for it, and you can have a small micro reactions are being made now to optimize the conditions. It provides the absolute quantification with each high specificity or high sensitivity. Then we have a other type of PCR. These are all variant PCR where we can do some variations, and the idea is that to get these variation to provide the best possible product. So standard PCR works up to 4 kilo-base pair, 2 to 4 kilo-base pairs, and or even more base pair can be optimized depending upon the template DNA and the type of tech polymerases or type of polymerases which are used for this reaction. The major challenges for this PCR, the primer miss annealing. So it can have non-specific binding. It could have like a secondary structure. DNA damage may take place because of long-range PCR, and proofreading polymerases are basically need to be present, and it's stabilizing the buffer, then optimized kits. So these are all solutions for that, and we can have the best possible reaction by optimization these reactions, and right from DNA quality of DNA to type of enzymes, DNTPs, even magnesium chloride is important. Single cell PCR works with the DNA from a single cell. Maybe colony PCR, you can have requires high sensitivity and specificity. You need not to have kind of a mixture. So basically, sometimes there's a contamination of is very important, whereas PCR get contaminated, reaction get contaminated with other DNA. So suppose sometimes you have a handling, and mishandling might cause like a DNA from different bacteria to get in, and you may have some some bacteria might present and get boiled, and after get lysins take place. You can have a DNA from some viruses and all. So you have always chance of some nucleotide bases incorporated the reaction mixtures where you have non-specific binding of the primers randomly takes place, and that reduce the sensitivity and specificity in order to get the product. So cells isolated by FACS are micromanipulation. specialized kit available. So these are the very specified single cells are taking. So you have a possibility of the contaminations or contaminant cells is totally negligible. And simply you take a cell, lyse it, and get the DNA from the single cell and proceed for the PCR. Now, the first cycle PCR called reduced runtime. Sometimes you can reduce the runtime. It also depends the size of the gene and the processivity of the tech polymerase or Vent or PFU or any kind of polymerases which are used for the PCR. Using fast ramping cycle. So fast ramping cycle is also depends on the machine. Sometime earlier, the Perkin-Elmer PCR, which used to be there in like 20 years ago or 25 years ago, they have a large ramping time. So it takes like 2 hours, 3 hours or even more, depending on your number of cycles and reaction time. But the ramping time may be reduced now with high technology, and you can have like in few seconds the temperature can be regulated in the machine, and you can reduce the time. For example, if you maintain 95 degrees centigrade, so from 95 degrees centigrade to an ambient temperature like a 50, reduced by 30 to 40 degrees centigrade, in very short time. So this is how you optimize the ramping time. Based on the ramping time, you can also optimize the reaction, optimize the reagents in that. Then short denaturation process again is important, because first in this case hot start PCR works well. We can have quick high boiling temperatures, short time DNA is taken without enzyme, and then you add the enzymes to increase to initiate the cycle process of the annealing and then process of denaturation, annealing, and extension. It maintains the specificity and sensitivity also. Now the methylation specific PCR we call MSP. For some research purpose and for the detection purpose, the DNA methylation after bisulfite treatment taking place, and this DNA after methylation can be used. 2 primer set of primers can be used. So methylated versus unmethylated. So you can also differentiate the methylated and unmethylated product of the DNA through this PCR. It requires the stringent conditions due to reduced DNA complexity. So DNA complexities get reduced. Now then we have many different variation in the PCR, which we call the marker, and we call the typing methods, and we call the marker identification, gene marker identification, using different type of PCR. Though the PCR reaction remains same, but we named them as in different. For example, RAPD, random amplified polymorphic DNA, where you have to have the short base pair sequences, which are random primers are designed, and these produce the unique banding patterns and profile from different individuals, from different microorganisms. Say, for example, multiple bacterial species or strains are used to get the DNA, and using this rapid method. So wherever these primer sites are present, they will amplify. So and then you can get the different patterns of banding. So this is basically mostly used for the typing method, and used for the DNA denaturation, DNA typing or marker identification method in the genome. Then again we have another type of we call rapid amplification cDNA ends, we call races. So these are all different names of the same type of PCR techniques used to give the different product. So variant of real-time PCR to obtain the full length cDNA. 5' race amplifies 5' ends, and 3' race amplified 3' ends. So when the 5' ends and 3' ends get amplified, the anchor sites are used for extension, and you will get these final products amplified on the gel to check. In situ PCR, the PCR performed inside the fixed fresh cell on slides. So this is how in situ technology combine PCR sensitivity with in situ hybridization. It detect, locate cell-specific DNA or RNA sequences. Then we have a differential display PCR, where we have RT-PCR based, the methods for comparing the gene expression. Use anchor primers for again for poly A tails, bending patterns compared across the samples, and now mostly replaced by the RNA sequence or RT. So RNA are also used for that, and quantitative real-time PCR or the PCR is used for the display purpose. So these are the techniques to analyze the PCR products. So this is the agarose gel electrophoresis, where the gel electrophoresis is being run. Say tests, then 1, 2, 3. So you should always have positive and negative control. Now, positive and negative control is important because positive control give you the idea that you have the gene present of the same size, same place, same location, and negative control will tell you the possibility whether you have something wrong, your reaction is working well. Suppose if you are getting the product in the negative control also, where there's a negative control means there's a no template, no DNA. Everything is there, all DNTPs, all primers, DNA polymerase, water, buffer, and everything. But you are not getting the template. So if even having no template, if you're getting band, it means something wrong. It means you have some contamination. So if you're getting the band in negative and band in the test sample both, it means your system, your PCR is wrong. It's not working well. Now for that you have to avoid all possibility of contaminations, right from primers to template and all. Then you have the positive control. Positive control will tell you yes, your DNA is correct and you have the right band. If your positive control works and your test sample doesn't work, it means your reaction could not work. So these are some rule out the possibility of whether reaction is working. A reaction is working but with contamination. So gel electrophoresis of PCR product, then cloning of PCR product, then you have a sequencing of PCR product. So all these things can be done to verify, to confirm the right product being produced through PCR. Now we have applications of PCR. Again, you have a number of applications of PCR. For example, genetic disease diagnosis. So you can detect the mutation like thalassemia, human globin disease, gene disorder. Then you have several SNPs can be detected. So you can have several mutations can be identified. So how you identify these mutations? You amplify the product and you get a sequence. So you compare the sequence of the control and the amplified product if it is mutated. If you find the variation in the gel. Now here one thing should be taken into care that whenever you talk about the mutation detection, you should be careful about the which enzymes are used for the DNA polymerase, because in case of tech polymerase, they don't have error proof activity. So there is always chance of mutation if you are using the long size gene. Say for example, if you are taking more than 2 or 3 kb or 4 kb gene, in 1 kb gene the possibility of mutation is very less. But for large size of a gene, yes, possibilities is increased, probability is increased, and so you may have some mutation. So in such cases, when you have large genes to be amplified, then you need to have the different high proofreading enzymes like PFU and Vent polymerases or some other polymerases. Variants of tech polymerases are involved to get the right product. So genetic disease diagnosis can be identified. Infectious disease detections, which viral particles are there, which bacteria, microbacterium, virus, COVID-19 virus, or HIV virus, HBV virus, which virus genome you have in the blood infections or in the cell infections, that can be detected through PCR. Forensic science analyze the DNA from hair, blood, so you can have any crime site can be used the PCR. The high sensitivity can amplify from as little as 1 DNA molecule. Forensic science uses DNA fingerprinting through these methods, and then paternity testings and crime scene, vagara. Now, the research biotechnology, in biotechnology and genetic engineering, the PCR is very highly useful. So, for example, gene cloning. So, in gene cloning, it's very difficult to get the gene from the genome by cutting the enzyme and get the product purified and use this product for the gene cloning. What is the disadvantage in that kind of a gene cloning? It is easy to say you cut the gene, take it out, clone into the another vectors and transfer into the bacteria and you clone the gene. Ideally, it is very simple. Just like 2 hours, in 2-hour reaction, you cut the genes and get it out by running the reaction. But do you really have the correct enzymes which are present in the gene that need to be cloned using same enzymes in the vector? So you are bound to identify a specific enzyme site what are present in your cloning vector or polylinker site, and they are limited, 10 to 12 sites. So suppose if your enzyme, that cloning vector site enzyme is not present in the gene or present in between the gene, you cannot take it, right? So you have to be smart enough to design a primers having linked with the restriction digestion site, right? For that, you need to use the cloning PCR. So you get primer, to get PCR, like you increase the PCR product having the overhanging regions of the enzyme sites and clone into the, so this is a targeted enzyme you can clone in that. Now, then mutagenesis. I know mutagenesis and transcriptome analysis with RT-PCR, PCR is very important. Reactions and very important, sorry, experiments which are used in the research of biotechnology. Site-directed mutagenesis, without PCR you can't do it. So you must have primers with slight mutations in the product in the templates, so you can increase the products and the product will amplified with that mutation, and that mutant product is cloned into the vector to give you the specific site specific mutation. Then microbiome and metagenomics. So again, 16S RNA, PCR, shotgun sequencing preparations, even back-end YAC. So you need to have a PCR product to amplify for the library preparation. In agriculture, agriculture and food pathogens detections in the plant. So you can also use the PCR method for detection of pathogen in the food, agriculture, or looking the markers whether they have been, for example, in transgenic plants. It has very highly used in the transgenic plants. Genetically modified product. So you can have GMO testing, like genetically modified organism testing in a crop and food products. Then authentication of a meat and dairy. So again, to look into the purity and quality control in the dairy products and the meat product, looking the specific toxin and pathogenic bacteria or the food poisoning bacteria, whether they are present or not. So simply amplify through the gene. What are the limitations of PCR? Every technique or everything that we either develop or we discover should have, besides a lot of merits, this should have some demerits. So the PCR is also having some demerits. Highly sensitive, prone to contamination. So wherever you have contaminant DNA, it will give you the false positivity, right? Suppose if you are testing the presence of HIV infection in a patient, suspected patient, you may have some false positive. You may get the positive results and you can say, okay, this patient have HIV infection. But really, he or she may not have HIV infection. So that had to be ruled out through some cross-check reactions, cross-check through sequencing, through some other methods. So it's a highly sensitive, and the sensitivity will give you the disadvantage that it may also bind some false site and give you the false positive reaction. It requires the prior knowledge of DNA sequence. So without having prior knowledge of DNA sequence, you cannot design a primers. This is one of the conditions, challenge or limitation. Non-specific amplification, primer, dimer, off-target product formation. So it may have this because ATGC, sometimes repeats are present. So that repeats might have, bind with the off-target. Then limited product size. So for a specific and for efficient PCR, you have up to 3 kb. But if you increase the size like a 20 kb amplicon or 50 kb amplicon, then that's one of the limitations. For that, you need to have more refined and high quality enzymes, which are expensive as well and may not have high efficiency to get this large product. Conventional PCR is not quantitative. For that, you need to go for the real-time PCR, digital PCR. Error-prone polymerase is another problem. For tech polymerase, you cannot have, you may have, have chance of mutation even in the 1 kb gene. So you cannot clone that with certainty, that product. That's the one limitation. Inhibitors in clinical soil food samples can block the, so there's a lot of inhibitors are present. Some unknown things are present, chemicals traces are present, metal traces are present in these products. So if you go for direct taking the DNA from soil, food and other, then you have a chance of some intervening between the enzyme and these products, and that give you the false, may not give you the correct results, may not give you the product. So with this I take permission and thank you very much for listening, and this is the, you can go to the T.A. Brown books. It's a very, very good book. I recommend this book, although it's a old version, but you can go with the new version. Even Ville has approached me also for the new editions of the Indian version of the T.A. Brown, where I also added some chapters. I don't remember, but I think I have added something on genomic based on PCR, like functional genomics part. So that can be read by you, and that give you big information about the PCR, cloning, and all kind of molecular biology technique. With this, thank you very much.